So I just want to give a brief introduction about Dr. Harshita Tiwari. Dr. Harshita Tiwari has a strong educational background and extensive research experience in the field of pharmaceutical sciences with a focus on computational drug discovery and molecular modeling. She worked as a senior research fellow at the Discovery Informatics Lab, Natural Product and Medicinal Chemistry Division of CSIR, Indian Institute of Integrative Medicine, located in Jammu. Dr. Harshita's dedication to her research and her contribution to various projects have earned her recognition and respect in the scientific community. Currently, Dr. Tiwari is an accomplished computational chemist at Celaris Therapeutic, Austria, and computational research scientist at Drug Chemistry Research Center. Dr. Harshita's educational journey includes a Bachelor of Pharmacy degree from Kumaon University and Master's degree in Pharmacoinformatics from the National Institute of Pharmaceutical Education and Research in Kolkata. She is also on the verge of completing her PhD under the guidance of Dr. Amit Nargotra, principal scientist at CSIR IIM Jammu, where her research has primarily concentrated on utilizing computational methods to identify and elucidate the mode of inhibition of potential inhibitors of E. coli, UDP, and acetylglucosamine enol pyruvyl transferase MUR-A enzyme. Her work extends to several other areas, including drug repositioning for neuroinflammation, molecular mechanisms of anti-inflammatory compounds, and database development. Dr. Harshita Tiwari's research contributions are evident through her numerous publications, and her expertise includes molecular docking, molecular dynamic simulations, pharmacophore modeling, artificial intelligence, machine learning, and data analysis. She has also actively participated in conferences, workshops, and training programs related to drug discovery and computational biology. Her dedication to advancing pharmaceutical research and her accomplishments make her a prominent figure in the field of pharmaceutical sciences and informatics. So we all welcome you, ma'am, for the today's webinar on revolutionizing drug discovery, the power of machine learning. So over to you, ma'am, and thank you so much for joining us.
Thank you, sir, for this warm welcome. Let me share my screen first.
Yeah, sure, ma'am. Now you are the host. You can present your slides.
Yeah. Can you see my slides?
Yes, ma'am.
Okay. So thank you very much, sir, for having me in this workshop, in this conference. So the topic of my today's webinar is revolutionizing drug discovery, the power of machine learning. So let me briefly introduce myself. My name is Dr. Harshita Tiwari. I am working as a computational research scientist in Drug Chemistry Research Center, dcrc lab. The flow of the webinar will be like this. So first I will introduce you with the basic artificial intelligence methods. Then I will quickly move to applications of AI at various stages of drug discovery research, majorly focusing on early phase drug discovery. Then I will conclude the important with telling you about the important AI based tools for drug discovery. Let's move toward our first session. So, by definition, artificial intelligence is a field which aims to provide intelligence to machine, which means that by the application of the artificial intelligence, we can train our machine for making predictions or for the classification related tasks. So, the field of artificial intelligence, it basically compasses of two parts. One is machine learning, which applies the statistical learning or statistical equations to achieve the task of artificial intelligence, and then the deep learning, which is a subset of machine learning techniques based on the deep neural networks for mimicking the behavior of the human brain. Then it can perform two major tasks. One is the classification task and one is the regression task. So if I'll give you an example, the classification task is the task by whose output is which gives the binary output. Like it can give output in the form of 0 1, yes or no. So suppose there is a student which has appeared in an exam, and we need to have a classification task. So how would we do that? We will simply classify, we will see its marks, and we will classify either he is pass or fail. Then, if you want to perform a regression task, then what we will look, we will see what is the percentage of marks. So, the thing is, if we perform the classification task, the output is always binary. And when we perform the regression task, we get a comparative output. Then it can perform the clustering task as well. Suppose we have a class of students which have different properties. Some are good in arts, some are good in music, some are good in maths, or some can be good in science. So with the application of the clustering, we can classify the students which are good in arts, which are good in maths, or which are good in science. So this is basically the grouping of the student based on particular traits. So till now we have learned that machine learning can perform three kind of tasks. One is classification, another is regression, and third is the clustering. Then it can perform these tasks by three types of learning. The first two are supervised and the unsupervised learning. So supervised learning is the learning when we have a label data. So what is the label? It means that we have simply named the output of some we have input data. Like I have shown here the few apples as an input. So here I have annotated these fruits like the apples as an apple. So I will say the model that if a fruit is red, it has a leaf, and it has a little pedicle attached to it. Then it it can be an apple. So I have trained the data with the labels. So this kind of learning, when we have a label data or name data, it is called the supervised learning. Then the unsupervised learning. Suppose I have given a data set of fruits to a machine learning model. So here in my data set, there are some apples, some bananas, and some peaches. So I have not tell my model which fruit is apple, which is banana, and which is peach. But I want my machine learning model to look at these fruits and classify itself the fruit as in three different categories. Then this type of learning will call the unsupervised learning. So how it will do that? It will simply look at the leaves first. So it will look at the apples, peaches, and the banana. Since banana has not leaf attached to it, so it will remove it from the category in this category. So it will group it as another subset. Okay, then it will look at the apple and the peaches. Then it will look that both have leaf, both have some kind of round shape. Then it will say that then it will move toward this task, and it will look at the color of the fruit. So it will say that there are some fruit which are bright red in color. They have one category, and some fruits which appears as an orange in color. So it will classify those fruits in the different category. So this is called the unsupervised learning. When we don't tell our data, we don't give any label to our data. Then the third kind of learning is a reinforcement learning. So here I have added an example of a baby. Suppose you have a little baby in your house, and he has freedom to move any part of your house. So what he will do, he can move towards the fire or any dangerous situation, or he can move towards the food. So whenever a baby goes towards the milk water or towards the food, he had some good experience. Okay, or you can say it adds some reward to the baby, and it learns that if he will go towards the right side, he will get some food. Similarly, if he chooses to go toward the fire, okay, or any dangerous situation, what if it will learn? It will learn that whenever he will move towards the fire or dangerous situation, he is harming to himself. Okay, so he will add a negative impact or negative rewards to the baby's mind, and he will not go towards that side. So this kind of learning, when a person is learning or a model is learning from its experience, or it is adding some reward to the positive situation and some negative rewards to its the negative outcomes. This kind of learning is known as the reinforcement learning. Okay. So what is we in the reinforcement learning, where we provide the input data, some raw data to the model, then we add some reward. Okay. So based on the rewards, we provide the output. So this is the third kind of the learning. So now we have learned that there are three kind of learning task here. One is supervised, second is unsupervised, and third is the reinforcement learning. So reinforcement learning is basically takes important role while we are dealing with some robotics. Then the deep learning. So deep learning is the part of the machine learning or the subset of machine learning which mimics the behavior of the human brain. So as we know that human brain consists of the neurons, which have some dendrites, cell body, axons, and synaptic terminals. So these elements of a human neurons help it to interact with some other neurons in the brain, because brain is a very complex structure consisted of millions of the uncountable number of the neurons. So these structures of the neurons helps neurons to interact with each other. Similarly, the artificial neural networks or the deep learning methods consisted of several input layers, which we call the neurons, which interacts with each other and provide the output. So this kind of structure is known as the artificial neural network. So it is able to learn without human supervision, drawing from the data that both unstructured and the unlabeled data. Then the soul of the machine learning model or the artificial intelligence. So the basically the all the machine learning models are depends on the data. So if we will provide a good quality data to our model while training, it will resulted in a good quality of the model. So whenever we provide a data to a model, it should be accurate, complete. It should be in a uniform format, consistent. It shouldn't have any duplicate, and it should be integrated. So if you will provide a good data to your model, then it will result in an accurate results. Then there are basically 7 steps of the machine learning. First is the gathering of the data. Second is the preparation of the data, in which we clean our data by removing the duplicates or fill the missing values. Then once we have a good quality of the data, then we choose a machine learning algorithms to train our model. Once we have trained our model with the provided data, then we evaluate our model with respect to the accuracy and its predictability. And if we don't get the accurate results, then what we do, we perform the hyperparameter tuning. So hyperparameter tuning is nothing but it is the set of some parameters on which the performance of the model depends. So we just uh, alter the parameters of our model, and then check the predictability of our model. Okay, once we got some accurate model, then we use that model for the prediction. So there are various types of the machine learning models, and which has application in different fields. So we can use it's for the diagnosis purpose, for image classification, for recommendation system, or for a game skill acquisition. So different kind of learning has different application, and it has got the wide range of application in today's context. So this was about a basic introduction of machine learning techniques in general. Now I'm moving towards the application of AI at various stages of drug discovery research, and I will majorly focus on the early phase drug discovery. Now, let me give you a brief introduction of drug discovery process. So it is basically the three, it can be divided basically in the 3 steps. One is the drug discovery, which is early phase drug discovery, then the clinical testing, and then the approval for the production. So the drug first phase of the drug discovery, it is basically consisted of target identification, lead discovery, lead optimization, and the preclinical testing. Then the second phase of drug discovery is the clinical testing in the human beings. Then once the drug has we have the results from the clinical testing, we provide the results for the approval for to FDA or EMA. And once the drug is approved or in market, we perform some surveillance on the drug's performance. We check if the drug is having some side effects or not, or if it is performing any good, it is providing good results to it. So this is the overall process of the drug discovery, and it is a very long and time consuming process, and it is very costly too. So whenever we are performing the computational drug discovery or artificial intelligence drug based drug discovery, our aim is to reduce the cost as well as the time in the process of the drug discovery. Then this process of the computational drug discovery can be divided into basically two parts. One is the bioinformatics and another is the cheminformatics. So bioinformatics is the process by which we are gathering the data from the biological targets, so which can be proteins, DNA, RNA. Okay. Then the cheminformatics is the process in which we are getting the information from the large chemical space or the compounds. So there are different compound repositories which are freely available as well as purchasable, which help us to perform this cheminformatics task. So for a successful drug discovery processes, we have to take both the processes together. Then, in context of the drug discovery, AI refers to the utilization of the computational algorithms and machine learning techniques to analyze the vast amount of data, identify patterns, and make predictions. Okay, so from where this large amount of data comes from to build the machine learning models for the drug discovery. So, this drug discovery. So, before moving towards this, I would like to quickly um, take you towards the different task of the machine learning I have told you, with context to the with context to the drug discovery. So I have told you about the classification task with respect to the classification of fruits. And now, I want to now in context to the drug discovery, suppose I want to classify the a set of kinase inhibitors. So if I need to classify this model, and I have a data set of compounds consisting of kinase activators and the kinase inhibitors. So what I will do, I'll train my model. It can be supervised or unsupervised with this data set, and I will simply, I will after training, I will provide the unknown data set, and it will tell uh, if whether these compound is a kinase inhibitor or not. Okay, so this is the classification task. Then the regression task, as I have told you, the regression task is the task by which the output is a continuous number. Okay, so here if in context to the drug discovery, how if I want to perform the regression task, what I will do, I will provide the data set of compound with the set of some values. Okay, here I have provided the log POW values or log P values of the compounds, like 1.77, 2.1, 2.2, 1.64. Okay, when I am providing the compounds with these values, and now I am training my model, and I am building a model and use it for the prediction of certain log P value. Okay, then it comes under the regression task, by which we are deriving the value of log P. Okay, and this is the regression task. Now, if I want to perform the clustering task with the respect of this, what I'll do, I simply provide the set of kinase inhibitor, and I don't have labeled these compounds with any activity. Okay, so I don't know whether this is a kinase inhibitor or not. Okay, then what the model will do, it will simply look at the scaffold of these compounds, and then it will cluster the compounds based on their scaffold. So this is the classification example with the unsupervised learning. Then the application of reinforcement learning in the field of the drug discovery. Okay, so suppose I have some compound. Okay, some scaffolds like this is the cyclohexene ring or cyclohexene ring with some methyl group or the oxygen ring attached, atom attached to it. Okay, so here the agent is the scaffold. So I have this scaffolds of the compounds. Now what I can do, I can either add some scaffold to the compound, remove or replace the atom. Okay, so each time whenever I will add, remove or replace the atom, it will have some effect on the activity of the compounds. Okay, so here if it will increase the affinity, so the reward here is if the affinity is increased, then I'll say this addition is the good. Okay, or if any addition decreases the affinity, when what I'll say it is not good for the new molecule. Okay, so what I will do, I'll simply add, remove, or replace some atoms in the molecules, and then I'll check which function is increasing the property. So based on this, I will just derive or generate a new molecule for a certain particular targets. Okay, so this task in which I am using a particular reward or the penalty function, this is the example of reinforcement learning in the field of drug discovery. I hope you might have understand this concept. Now, as I have told you, there are 7 steps of the machine learning. So, in context of the drug discovery, the first step is the gathering and preparation of the data. So, in the drug discovery, the data comes from various experimental as an outcome of the various experiments. So it can come from the scientific literature which the scientist has reported from their previous experiments. It can be the patents related data. It can be the data related to the clinical trials in which a compound has failed or it has got approval for a particular kind of activity. It can come from the marketed drug. Either the kind of drug has performed well or it has created the side effects. Then the small molecule crystallographic data, like the data might have come from the crystallographic experiments or the or the experimentation techniques. Then the protein ligand complexes related data. So this kind of data might have come from the docking experiments, from the crystallographic experiments, from different which is stored in the different databases like protein data banks. It can come from the scaffold or the isostereos. Like we have performed a particular pharmacophore based assessment of the compounds or the QSAR based assessment of the compounds. We have performed the bioisosteric replacement of the groups in particular compounds. Okay, from this, the different kind of data we gather to build the models for the drug discovery. Okay, so now the data is in raw form. Our task is to remove or to prepare the data for the building the models. Then once we have set of compounds from these data sets, we have to calculate the molecular descriptor of the compounds. So in general language, these are some properties of the compounds which describe the compounds. So molecular descriptors can be 1D, 2D, 3D. So in terms of the 1D descriptor, it just tells about the elemental composition of the molecule. For the 2D descriptors, it tells about the number of hydrogen bonds, number of hydrogen bond acceptor, log P values of the compounds. And in terms of the 3D descriptors, it tells about the properties which basically depends on the 3D structure of the compounds. So they can be molar surface area, molecular volume, etc. So these are the molecular descriptors we need to calculate from the prepared data set, from the collected data set of the compounds. Then once we have these molecular descriptors, like here's an example of development of ML, a model of solubility prediction of the compounds. I have the training set of the compounds which have some solubility value. So I need to calculate the solubility of the compounds um, of the compounds. So these features are known as the molecular descriptors. So I have calculated the molecular descriptors, that is molecular weight, hydrogen bond acceptor, hydrogen bond donor, and the ring feature of these compounds. Okay, so it as you can see that the for the molecule number 1, the molecular weight is 300. For molecule number 2, the molecular weight is 450, and so on. Okay, so you can see that the solubility profile of these molecules, it is also changing. Okay, as the molecular weight is changing. Okay, similarly, the solubility value of the compounds or the molecules are also changing as the hydrogen bonds are changing. Okay, same in the case of the hydrogen bond donor. But when you see in case of the ring character, okay, either the solubility value is 1.23 or 1.55 or 2.0, 0.3. The ring number of ring is not changing. So we can say that the property means the property which we need to predict like solubility, it is not depending on this ring feature. Okay, so we can remove this feature to minimize the errors while developing the model. So this process of removing this unwanted feature while building the model is known as the data cleaning or data processing. Okay. Now we will build the model of solubility prediction with the help of the three features which are important for the solubility predictions. Now, what we have done till now, we have collected the data set of compounds, then we have calculated some descriptors, and we have removed the descriptors or the feature on which the solubility value is not changing or not depending. Okay, now we can build the model, then evaluate the model. Okay, now you can see that I have divided the data set for the model building in two sets, like one is the training set and another is the test set. Okay, so training set is the data set of the compounds which we used for the building of the model, training of the model. Once we have built the model, we can use the test set for evaluation of that model. So this is the overall example of development of a model with the help of machine learning. Then the application part of the machine learning. Okay, so it has a wide application from target identification and validation, from compound screening and lead discovery, from preclinical development to clinical development. Okay, so for each task we need different kind of data. Then this is a landscape of the companies which are working or using these drug discovery of machine learning or artificial intelligence technology for different tasks. So the first one are the companies which are using these techniques for early phase drug development. The second phase companies are using the AI based models for the preclinical development, and the third group of companies, they are using the AI based models for drug for clinical development. In addition to that, some of the companies, and they are the very big pharmaceutical players, they are using the AI based techniques for end to end drug discovery development. Means they are using the AI based drug tools for all these three processes. Now, some of the breakthroughs in the field of the AI based drug discovery. So there is a company which is known as the AI in silico medicine. So they have used their generative AI. So generative means they are using they are using reinforcement learning for the generation of the molecules. Okay, so in 46 days they have generated and a compound with the help of these AI techniques in 46 days, and they have checked those compound in vivo models as well, and the compound was active for the cancer. Then another breakthrough is DeepMind's AlphaFold. As we know that protein folding is a very important task because the structure of the protein depends on the structure of the function of the protein depends on the overall structure of the protein. Okay, so the DeepMind is a company which is using these artificial intelligence or the deep learning tools for the drug discovery purposes. They have designed a tool, AlphaFold, which can predict the the structure of the protein with 90% accuracy. Then another breakthrough is the first breakthrough was the by the Extensia. So it is the company which is using the end-to-end AI techniques for the drug discovery. So they have announced their first ever AI designed drug molecule to clinical trial in early 2020. Then in July 2021, the DeepMind announces the AlphaFold model to predict the 3D structure of the protein. Then in February 2022, in Silico Medicine, they started the clinical trial for the AI based developed drug, and in February 2030. So this drug, which in silico medicine has brought to the clinical trial, it got the orphan drug status. Okay. Then another breakthrough is in January 2023, AB Science, they have designed an de novo antibody. So de novo means from the scratch. Okay, so they have developed the de novo antibody and validated it in validated is. So they are using generative AI for this purposes. Okay, then this is the breakthrough for the related to the AlphaFold. Then the recent example or the recent breakthrough is the alpha missense, which is again proposed by Google's DeepMind. So what they have done. They have they have created a model which can predict the missense or the mutation which can lead to the pathogenicity of which can lead to the pathogenicity. So what will be the future perspective of utility of this model? Suppose as we all have suffered from the COVID pandemic, okay, in future we also encounter some new pathogens, and we know and nothing about these pathogens. Then and we have such tools like alpha missense, so we can check the genome of this new pathogens, and we can predict which pathogens means which kind of mutation it can have in the future, and and which mutations can be pathogenic. So we can design the antibiotics or the vaccines in advance to avoid any fatalities in future. So these are the some breakthroughs of the AI in the drug discovery. Now moving towards some important AI based tools for drug discovery. So now, as we know that the process of the drug discovery starts with the literature survey. So, and the literature survey, it is a very cumbersome task, and it should be very accurate, and it is impossible to go through all the papers and papers for designing your study. Okay, so there is a tool which is known as the Elicit. So it is an AI based tool which can if you will give, like I have provided the keyword EGFR here. It as soon as you hit the search button, it will give you all the papers which are related to EGFR, and it will provide the abstract summary of the paper, and it will also provide the interventions like what drugs are used in this paper, what are the outcome measures, like what is the IC50 of the compounds, what is the MIC50 of the compounds, okay, how many number of participants in this paper. So this tool is very useful and designed with the AI, which you can use for design your study and for the literature survey. Then another tool which you can use is the AlphaFold tool. So as you know that our body is made up of proteins as a macromolecules, and these proteins serve in different functions. So these proteins are represent a very good target for designing any novel drugs. Okay, and for designing or targeting any drug or the target, we need to have some the information related to its structure, because the structure depends on the protein. So the structures can be divided into primary sequence, then primary sequence structure can be divided into primary sequence, which is the arrangement of the amino acids in a string. Then the secondary structure, when these amino acids interact with each other by the help of hydrogen bonding, it gave rise to the secondary structure. Then the tertiary structure, which is the arrangement of the secondary structure in the 3D space. Then the quaternary structure, which is the arrangement of these tertiary structure for a functional form, like in the case of the hemoglobin. Okay, so this information is very important for targeting a molecule. So this it is it is easier to get the information related to this primary structure. But when comes to the tertiary and the quaternary structure, it is very difficult for some protein to derive this structure. Okay, so there are some experimental procedures by which we can derive the structure of the proteins. Some is X-ray diffraction, NMR, and the cryo-electron microscopy. But the major disadvantage of using this model or this method, experimental techniques, is that these are very difficult, which needs expertise, as well as it is very time consuming. Okay, so we cannot quickly give any protein sequence or protein to these methods and say and derive the structure quickly. Okay, in that case, like I have discussed earlier, that the structure is function depends on the sequence. We can use the some predictive softwares like iTester modeler, Swiss model, and now the AlphaFold tool, which is an AI based tool for the to get the information related to the protein structure. So the accuracy of this structure is 97%, and they have created a separate database of the structure which are derived from this AlphaFold tool. So if you are not getting any information related to target in protein data bank, you can query the structure in this AlphaFold database, and you can get the information related to the 3D structure of that protein. Okay, so this is very important tool for the drug discovery. Then the third is the Vega. So, as you know that for the development of these machine learning or AI based models, we need to have some expertise in the programming, okay, or expertise in the programming languages for the implementation of these tools. So, in that case, when a person doesn't have any experience or prior knowledge to the programming languages, they can use Vika. So this is basically a GUI based tool by University of Ricardo. So here you can provide your data. It can perform all the tasks related to machine learning, like data processing, pre-processing, cleaning, visualization, and simple machine learning model building. So these are the important tools for machine learning model building. Then some, if you have knowledge to the programming languages, then you can use a specialized library for machine learning model building. So these, the first one is the RDKit library, which is very important for cheminformatics related tasks. Here you can perform you can calculate the descriptors, you can perform the basic mathematical operations, you can visualize or convert the molecules, molecules to another library is the DeepChem library. So this is basically for the cheminformatic purposes. You can visualize your molecule, you can convert the molecules in different format, or you can also minimize your molecule with the help of DeepChem library. Now we have these library, and you want to implement these library, you can use the Anaconda software, which comprised of different programming platforms like R, Jupyter, for the implementation of or the building of your machine learning models. Okay, so these are some of the important tools which you can use for your research. Coming towards the conclusions. So as you know that these are there is an increasing demand of these tools in the field of drug discovery, and they have provided a very promising result to augment the overall drug discovery processes. As you can see that in silico medicines have generated and evaluated their molecule in 46 days. Then with the accessible framework, now uh, researchers there is no they have overly developed the tools for the drug discovery. Researchers needs to get involved more in testing, validated, so that robust tools can be developed. So there are different tools now available by which we can implement these methods. Okay, with this I would like to thank all the participants, and I hope this sessions will be informative for all of you. Thank you.
Thank you so much, ma'am, for sharing your experience with us. And I welcome all the participants to share their queries in the chat box, or you can directly ask by unmuting yourself. Machine learning is a very trending field of nowadays for basically for drug repurposing and drug discovery. So if you have any query, just let us know. There one query. It's a not a question. Ma'am, could you give in hand on training on those softwares? So definitely we are organizing time to time training programs. So hopefully in the coming week or month, we will organize a training session. Okay, in the machine learning based softwares or different softwares for drug discovery. So stay tuned with us, Mr. Gaurav. Okay, and any query from different participants who joined this webinar? Ma'am, I think you taught very well, so there are no queries.
Yeah, hello.
Yeah. Dr. Karthikeyan has a query. Yeah, yes, sir. Go ahead.
Yeah, thanks, Mr. Manu. Thank you, Dr. Harshita. It was a very, very informative session. I have one query. Can you elaborate more on the application of machine learning in target prediction in drug discovery
Yes, sir.
using
So
the
actually,
using the genomics data?
yes, sir. So now, as we know that genomics is it is a very old means it is very well studied subject. So we have lots of data from human genome sequencing, as well as the technologies are advancing. We are getting more data from the genomics experiments. So we can analyze these data with respect to the expression of certain genes. In particular phenotypic situation, like in particular disease situation or healthy situation, by which we can predict which genes are important for a disease progression, and which doesn't play any role in the disease progression. And we can also go for the precision based or the gene based drug discovery with the help of these machine learning data. So as I have mentioned extensia. So they are using these genomics based studies in their lab, and they are applying these methodologies for precision based medicine designs. So they are working very well in this field. This is the application part
Yeah.
of.
So
Yes.
one interesting area I would like to explore is the genes responsible for the development of resistance. And since you have good experience in antimicrobial drug discovery, for example, treatment of different organisms or different bacteria with antibiotics leads to the development of resistance. So, do you have? Is there any database which is available, which gives you information about the expression of genes after the treatment of antibiotics? Are those genes responsible for the development of resistance?
Sir, I don't know. I don't have any information related to the name of these, but the Google missense tool I just mentioned in my presentation. It might be helpful for this purpose because they have predicted this. They have used this tool to predict the pathogenic mutations or the treatment which leads to pathogenic and which doesn't have any role in the disease progression. So this tool can also be used for the studying the disease response or the resistance of the drugs in particular microbial infections too.
Okay, doctor. Thank you very much.
Thank you, sir.
Thank you so much, sir, for putting your query with us. And ma'am, we have a query from Mr. Sagar Rajput. Drug discovery had become more effective due to AI, but still it take lots of time in preclinical trials and clinical trials. Does AI can be also used in preclinical and clinical trials to make it more time effective?
I So, with the help of these AI techniques, we are minimizing time in early stages of development, right? In future, I believe that there will be some models which can mimic the behavior of animals too. I'm not sure about the humans, but for the animal behavior, they can mimic. So, we can reduce the time in the preclinical development of the drugs with the help of these AI-based tools.
Okay, ma'am, I think you are also presented in your slides that there are various companies who are working for clinical trials and preclinical. Okay, uh, in the last slides.
Yes,
So,
they are
so
working.
this is yeah, this is possible. I think already possible. Already many companies are working on it. So
Okay, sir.
we have another query from Mr. Gaurav. Ma'am, I have a doubt that what strategies are being developed to overcome the data security problem in drug discovery, analysis of a drug with machine learning, especially in rare disease.
Okay, so that was not data security. I guess this is data scarcity. Overcome the data scarcity
Scarcity, yeah.
problem in drug discovery analysis of drug with machine learning, especially in rare disease. So I don't think there is a data scarcity because lots of research has been done. So now the thing is that we need to develop some uh, accurate models and we need to work on the data cleaning. Okay, one thing is that. Second thing is that we have some positive data because most of the researchers they report the positive data, but we don't have negative data. Okay, so data imbalance is another aspect. So if we will start reporting our negative result as well, then the problem of the data imbalance that that can be solved. Then moving towards the problem of applying these methods in the rare diseases. So, if you look from the perspective of the companies, okay, so companies are rarely working in the means they really rarely work in the field of rare diseases because they don't get much profit from that. Okay, but I was talking to some of the people who are working in this area. So now people are means they are increasing the funding in the field of rare diseases. Eventually, the field of rare diseases based drug discovery will also bloom. So yeah, that was my answer for your question. I hope I have
Thank you
answered
so
your
much,
query.
ma'am. And is there any other queries from the participants? Okay, there are various renowned personalities have been joined our webinar, like Dr. PK Singor, sir. If you have any query or you want to share your experience with us, sir.
Uh, no, I'm just listening because I'm also learning about the machine learning. So Madam told very very good manner and very good way. I'm just ask one thing. I have one query that can there is some freely available software that regarding the artificial intelligence in the drug discovery. There is some freely available software or not?
Sir, most of the tools are free for the
Okay.
artificial intelligence. So if you have some knowledge related to programming, then you can go for the Python or Anaconda, and you can use the libraries like DeepCam and the RDKit for the implementation or building of the model. And if you don't have, you can use Vika. And for the and there are some tools for the docking studies as well, like Deep Doc. You can use for that for machine learning based docking studies as well. Yes,
Okay. Thank you. Thank
yes.
you, madam.
Thank you, sir. Sir, I think we cannot hear you, sir.
So, ma'am, there is no query from the participants. Okay, so now we are going to bind up the webinar, and I would like to welcome Dr. PK Singor sir for vote of thanks.
I would like to thank Dr. Harshita Tiwari, ma'am, for explaining well because I'm also working on drug designing, but I'm new in that machine learning and artificial intelligence. So I'm learning. I was listening very carefully, so I can then implement, and I will learn also from you when you will organize that hands-on training. So I think all the participants will get the knowledge, and then they will utilize the concept in future also. So I thank you very much, ma'am, for that giving the times to all to sharing this knowledge. And I thank to Mr. Monu Sukla sir for inviting me also. So thank you all very
Sir, I'm still your student. Don't call me sir.
much. No, no, no.
Thank you
Okay,
so
all
much,
is
sir.
sir. Okay, thank
Thank you so much, sir, for sharing your experience with us. So I would like to welcome Dr. Karthikeyan to give a vote of thanks and share your experience with us, sir.
Thanks, Mr. Monu. It was a great opportunity for me to listen to Singor sir and Dr. Harshita. And it was a very relevant topic and very up to date. So it's quite interesting to see so many students attending this seminar because drug discovery, as you know, it's a evolving discipline. So it is always good to be up to date with the recent developments in this area. And machine learning is a very interesting area, and in which I think our country has a lot of potential. So training the students in this very important area would be a great contribution to our country. Thank you once again, Monu and Dr. Arshita and Singor sir. Thank you.
Thank you so much,
Thank you.
sir. So it was not a national webinar; it was an international webinar. One of the participant from US, Mr. Subham Pandey, was here in the webinar. So I would like to welcome Mr. Subham Pandey to share your experience with us. Mr. Subham, are we audible for you? He is from Alabama University, from US. Okay, so there's a query from Subham Pandey. Thank you, ma'am. If possible, please arrange some workshop on training in machine learning. So I just want to tell every participant of this webinar that we are going to organize some training programs on CAD, computer-aided drug design, as well as machine learning for the drug discovery. Okay, so stay tuned with us. We are timely organizing webinars, training programs, and conferences. So you can time to time check our website, that is dcrc.co.in. Okay, so and we are also welcome to all the participants. You can contact us for collaborative work as well as as well as training sessions. So we will also organize some training session and webinars with your institutions and universities. Thank you so much, ma'am, and all the participants to share your experience. Okay, so ma'am, I would like to share one slide which is related to the certificate of appreciation. So uh, please make me host so that I will be able to present my slide. Okay, thank you
I
so
did
much,
mute
ma'am.
you.
Thank you so much, Dr. Harshita Tiwari. She is also working as a computational research scientist at dcrc labs, as well as she is computational chemist at Celaris Therapeutics. Thank you so much for sharing your experience with us. Hopefully this will webinar will be fruitful for everyone, and it's a trending topic for current drug discovery. So thank you so much, ma'am, and all the participants for joining the webinar. Thank you so much. The certificates and recording will be provided you within a week. Thank you so much.
Thank you.